Good morning, ladies and gentlemen. I just want to say thank you for your time, and especially James here, the CEO of Nyrada, to join us, have a very, very in-depth discussion of a few key topics that he wants to talk about with the business. I'm excited myself. I'm sure he is as well. And James, thank you again for your time. My name's Harry McCaskill. I'm one of the senior advisors here at ShareWise. We're a share advisory firm in Sydney. And like I said, we're looking forward to hearing from James, but I'll let James sort of introduce himself. A little bit of a presentation. He'll share his screen, sorry. And then, like I said, a Q&A at the end where we can answer any questions you guys might have. So feel free to drop in any questions throughout the conversation, and we'll go from there. But James, thank you again.
Yeah, thanks, Harry, and thanks ShareWise for the invitation. It's great to get the opportunity to tell the Nyrada story.
Excellent. I've been following for a little while, so I'm excited myself, but I'll let you take it away.
Okay, so Nyrada is an ASX listed biotech. We're based in Sydney, and we're a developer of small molecule drugs. And our lead program is a drug we're developing to treat brain injury. So I wanted to start by covering a little bit about my expertise and what got me to join Nyrada. So back in the early 2010s, I was involved in a major traumatic brain injury study. It was one of the largest studies run at the time. It was a 270 patient study run at 20 sites in the US, and we recruited around about 270 patients. And these were patients who were moderate to severely injured. One of the things that struck me was two things really. The frustration around the fact that there are no drugs that have been approved to treat this indication. So there was a sense of frustration not only from the families of these victims, many of whom were young people who'd suffered serious motor vehicle accidents or falls, but also from the medical professionals that I was dealing with. And I remember a story a trauma neurosurgeon told me at a site in Texas. He was saying that even a drug that gave a 10% functional improvement would be revolutionary. So that really stuck with me. And, alongside the fact that it was a poignant reminder of the seriousness of TBI for patients and these families. So what I'm going to do in this presentation, it's a short presentation. I'm going to run through the significance of this technology, how it works. I'll cover that briefly, and what our plans are, and why I see it as a compelling opportunity for investors. So in a nutshell, Nyrada's mission is to change the unacceptable reality of brain injury by developing a groundbreaking neuroprotective drug. And the aim really is to bring hope and tangible improvements to patients and their families. So here's a copy of our standard disclaimer. This can be found in the deck that we submitted to lodge with ASX this morning. So a little bit about Nyrada's expertise in the area and what our approach is. So I mentioned we're a developer of novel small molecule drugs. We use what's called a rational drug design approach, and that differs from what was done in the past, where biotechs and pharma companies would screen thousands of molecules to see what worked. With the advent of technology, we take a different approach. We select therapeutic targets that are interesting to us, and we develop novel molecules using sophisticated computer modeling. So the focus for the company is on developing novel treatments for serious and life-threatening conditions, and particularly in areas where the existing treatments are either suboptimal or even don't exist. And traumatic brain injury and stroke fit this criteria. So just a little bit about the origins of our drug program, our brain injury program. Back in the mid-2000s, and this was following on from advancements in gene editing, scientists were able to develop a mouse which had an ion channel knocked out. And these are TRPC ion channels, which is the target for our drug, and what I'll be talking a little bit more about. What this knockout mouse did was it enabled scientists to study the effect of these channels on the body. And what came out of that was TRPC channels being a promising therapeutic target, and that's what we went after. And one of the findings from this research was that these TRPC knockout mice, and this is a remarkable finding, they were protected from secondary brain injury. So that work, along with some pioneering work that was done at UNSW Sydney, led to the development of a drug, NYR BIO3, which is our lead compound, and that's what I'll be focusing on today. So there are some features of this program that are worth pointing out. The fact that we use rational drug design means that we designed a drug with one specific target and one specific purpose in mind, and that is to block these TRPC channels, effectively doing what is happening or occurring in the knockout mouse. And we were able to develop a molecule, NYR BIO3, that is not only highly potent, but just recently, back in February, we published data on an efficacy study in a stroke mouse study. And the result we got from that was quite astounding. We got a 42% neuroprotection in this mouse with our drug. So that showed that the drug worked, and it sort of validated the central hypothesis. We're also hopeful that the drug will be safe and well tolerated. The fact that these knockout mice who don't have these ion channels are viable, they live normal lifespans, they breed normally, and so on. And work we've done so far has confirmed that with our drug. But before we advance the program into the clinic, which is the next milestone for the company, one thing we have to do is prove that the drug is safe and tolerated in a battery of studies, and that work is underway now, and we're reporting those results as they come in. So that's what we have to do before we advance to the next stage. One of the other things I'll point out about this program is the fact that TRPC channels, based on this research that was done in the knockout mouse, has been shown to be a therapeutic area that covers multiple indications, even outside neurology. So these TRPC channels are widely distributed or expressed in the body. So they play a role in a number of diseases outside of stroke and traumatic brain injury, including in therapeutic areas like the cardiovascular system, kidney diseases, as well as cancer. So the strategic focus of the company currently is to focus on neuroprotection with NYR BIO3. And what we're looking to do is specifically target moderate to severe traumatic brain injury and stroke. So they're the first indications that we're going after. So just a little bit about the market. The TBI market is large, as is the stroke market, and they represent unmet clinical need. So there are currently no approved drugs for treating traumatic brain injury, and the treatment options for stroke are rather limited. In the US, each year around 2.5 to 3 million TBIs occur. And of those, about 10 to 20% are moderate to severe. So we're talking about a lot of patients, 250 to 600,000 patients who are underserved at the present time. So the stroke market is, in fact, even larger than the TBI market. Each year in the US, around 795,000 people suffer a stroke. About 85% of those are ischemic strokes, and I'll talk a little bit more about that shortly. The only treatment option available for ischemic stroke is a drug called it's a tissue plasminogen activator, or TPA drug, and what that does is it breaks up the clot in an ischemic stroke. Unfortunately, the drug is only applicable to about 10 to 12% of patients. The reason for that is that it needs to be given within a very strict 4.5-hour window, and a lot of patients don't make it to get treated within that time period. So there's a large percentage of patients who are underserved, again, in the stroke market. In terms of the size of the TPA market, just to give you some context, the global TPA market each year is worth between around 1.5 to 2 billion US dollars, and about 50 to 60% of that revenue is generated in the US. So just one other point I'll make on this slide, we've been successful in establishing a number of key collaborations. We've got a collaboration with UNSW, and we're very fortunate to have on our scientific advisory board Professor Gary Housley, who did a lot of that pioneering work with the knockout mouse. And we also have a formal established partnership with the US Department of Defense through the Walter Reed Army Institute of Research. That's very important to the company because they ultimately will be one of the larger customers if we're successful in developing this drug, because TBI, as you would appreciate, is a major health concern in the military. Yep. So that collaboration is structured around a cooperation, research and development agreement, and we're hopeful that all going well with our development, that'll pave the way for non-dilutive funding for later development in TBI.
That was going to be a key question I had. What are the opportunities for non-dilutive funding for Nyrada, James?
Sure. Well, a lot of work has been done in the last decade since the trial I was involved in setting up infrastructure to run traumatic brain injury trials. And there's a work group that's been set up between the US Department of Defense, the FDA, and industry, as well as academia, to look at these questions around how these trials are run. So a lot of the funding has gone into setting up that infrastructure, but now there's a focus back onto funding promising drug treatment programs. And so I think we're in a good position. We're in the right place at the right time with this drug in this indication. And it's something we're actively pursuing.
Yep, excellent. Thank you.
So this slide here sort of explains what the drug does. And with brain injury, there's a primary injury that occurs immediately after the impact or the ischemia in the case of stroke. And with TBI, the injury is caused by either mechanical forces, and they can be penetrating or non-penetrating, or rapid and sudden acceleration or deceleration. And what that does is it causes an infarct to form as the primary injury. In stroke, the mechanism's slightly different. It's usually caused in the case of an ischemic stroke by a blood clot. And that causes oxygen deprivation, and you get an infarct. But secondary brain injury, which I'll come on to now, is common across both. Just in terms of the primary injury, which you can see in the blue panel at the top left, it typically affects about 5 to 15% of brain tissue, but that depends on the severity and location of the injury. Secondary brain injury is a process that occurs in the hours and days following the injury. And what this does is it expands the area of damage. And it can typically add an additional 25 to 40% depending on the severity of the injury. And you can see that in the green panel at the bottom left. This is actually a rat brain from a pilot study we did with Walter Reed. And you can see in the top image the primary injury, which this is a penetrating brain injury. It's the brown shaded area that runs through the brain. And the slightly lighter color in the green panel indicates secondary brain injury. So that's what this drug is designed to address. That secondary injury is caused by a process called excitotoxicity, which I'll come back and talk a little bit about shortly. But the graph on the right, it sort of summarizes the trajectory and the objectives of this treatment. What we're looking to do is minimize that secondary injury shown by the green line, from the solid line down to the dotted line. And the aim of that is to minimize the area of brain tissue damage and minimize the level of disability. So what the drug does is it blocks these TRPC channels, thereby lowering or mitigating excitotoxicity. And I'll talk a little bit more about that shortly. Now, this is a bit of a scary slide if you're not a cell biologist, but I've got an analogy here which might help. This image here represents the bio machinery behind neuronal signaling. And so what you can see here is the surface of a brain cell. And just as sort of an interesting aside, the average adult brain has 86 billion of these neurons. So quite a large number. And the way these neurons communicate is that they use chemicals. And one of the key chemicals for signaling is a chemical or a neurotransmitter called glutamate. And you can see that labeled there. And what that does is it binds to a receptor and it sends a message to the cell to basically get excited and start sending messages. And so one way it does that is by opening these ion channels, which you can see on the right-hand side, and you can see the calcium is the blue dots. Now, the problem with glutamate, in a normal set of circumstances in a healthy brain, that whole process works quite smoothly. But when a brain is injured or a neuron is injured, it starts releasing glutamate, and you get this compounding effect of too much glutamate opening these ion channels and letting in calcium, and it gets into an overexcited state. And one way you can think of this is the cell being like an electrical grid, with the neurons rely on a steady flow of calcium, but what happens following injury is that you get like an electrical surge, and that is damaging to the cells. So what NYR-BIO3 does is it effectively blocks these channels, like you would see in an animal that didn't have these channels, and that limits the excessive calcium entering the cell. So this calcium builds up when these channels open to levels that are toxic. So it effectively acts like a circuit breaker, and that mitigates the risk of secondary injury. So the company's got a very busy 6 to 12 months ahead. We've got several key catalysts that will really determine the trajectory of the program. I mentioned we're undertaking safety and tolerability studies before going into phase 1. That's progressing very well, and that that work is necessary before we can move into a phase 1 study. So we'll be reporting on that work as it proceeds, as we receive the reports. So watch out for that.
And James, just to jump in really quickly, we've had a question. Someone asking, are you fully funded for that human phase 1 clinical trial for the NYR-BIO3?
Well, in terms of funding, there are a few unknowns at the moment. We're still getting quotes for some of the key work. We're currently having our accounts audited, yep. And we'll be releasing that information to the market shortly, I believe in the next week or so.
Yep.
And also the other unknown is the R&D tax rebate. So we've got an R&D tax rebate that will be due for the 24 year. And we haven't calculated the quantum of that yet. So there are a few moving parts to this, but there'll be some news on that when we publish our accounts.
Excellent.
But we do, in the last accounts, we reported 4.7, just over that, $1 million in the bank.
Yep. Yep.
As I said, with the R&D tax rebate pending.
Yep.
So the other key milestone for the company in the near term is obviously the initiation of that phase 1 trial, and in parallel with that, we're also running this study with Walter Reed, looking at the drug in a penetrating ballistic brain injury model, which is along the lines of those images I showed earlier. So that'll provide crucial efficacy data to take forward the drug in TBI. So here you can see an overview of the study design. So it's a healthy volunteer study, and it's going to be run here in Australia. In fact, here in Sydney. And it's a two-stage design. So initially we'll be dosing healthy volunteer subjects with single doses, and there'll be 3 cohorts of those doses. They'll be escalating. And following that, we'll then be dosing for 72 hours in 2 later cohorts. Each cohort of subjects will be 8 in number. 2 will receive placebo and 6 will receive NYR-BIO3. And the objectives of the study, what we're trying to do here is to show that the drug is safe and well tolerated in healthy human volunteers. And what we're also trying to determine is what the pharmacokinetics, what the blood levels look like, how long the drug stays around in circulation, and so on. So just to summarize, I think the program presents multiple compelling reasons for excitement and optimism.
I think you went back 1 slide instead of forward.
Oh no, I'm just wrapping up. I did go back. Sorry.
Okay.
Yeah. So this is just to wrap it up. So the key features of the program, it's a first-in-class opportunity. This drug is novel, and it's treating unmet clinical need. The markets in stroke and TBI are large, and unfortunately they're growing. People are having strokes younger these days, and also we've got an aging population. And on top of that, in the TBI space, we've got global conflicts around the world as well, leading to more TBI injuries. As we discussed, or as I mentioned, there are non-dilutive funding opportunities available, particularly for TBI through the DoD, and that'll enable us to secure funding if we're successful without shareholder dilution. And also, I think the final thing to say there is that we're pioneering in an area with a promising target and a potent drug. So that opens the way for us to move into other indications, and that'll drive growth for the company.
Yeah, excellent. I think it all looks very, very good. We've got a couple of questions coming through, so
sure.
we may as well answer a few of those
Yep.
and see what you've got to say. So we've just had one come through from Colin. What is the potential? Sorry, James. What is the potential for treatment of concussion, for example, in sports injuries and things like that?
That's a great question. So concussion it comes at the mild end of the TBI spectrum. We've made the decision to go after moderate to severe initially because we think that's where the urgent need is, and we see it as quite a different market. Yep. The patients that are moderate to severe are hospitalized, so a 3-day IV treatment is suitable for them, but that wouldn't work for a concussion patient. The concussion market is very large, and it's one we're definitely going to come back to and have a closer look at, but it's not the priority for now.
Okay, excellent. I think you want to focus on the core rather than the little things first, and then obviously that can come down the track. And then with regards to your approach and the science behind NYR, what's the difference, or what's the comparisons to AGN? So the AGN drug, it's a peptide, yep. And that works on a different target. It's hard to draw comparisons. The objective with their drug is the same as ours, but it's the science behind it is different and distinct from Nyrada's. Okay, excellent. And another one, we've got one from John. Can the phase 1 study support other indications for NYR-B103? Absolutely, yes. Okay. So any indication where the drug can be delivered intravenously, the phase 1 study would support that. Excellent. But not for oral administration. That would be a separate study. Okay, right. And I guess for yourself, obviously you've got a lot of experience in this industry, in I guess the company within Nyrada. What's in the next 5 years, where can you see the outlook heading? So following the completion of the phase 1 study, we're going to open an IND, investigational new drug application with the FDA. Yep.
And we see later development being split between Australia, because we want to keep a base here, but also the US. And from previous experience, it just gives us access to a larger patient population, a patient pool.
Yep.
So you can run trials more efficiently, bring on more sites. Because one thing we can't buy is time. So you want to run studies as quickly as possible.
Okay, and again, they do take time, but you obviously can't rush them. So you've got to be patient with those sort of things. Well,
just further to close out that question, in terms of where the pathway for the TBI program, I think there's a favourable regulatory pathway for that drug.
Right.
For the drug in that indication, and the reason for that is it's a serious and life-threatening condition, and it's unmet need. So, we're looking potentially, not confirmed, but smaller studies, and maybe one pivotal phase 3 study. But that's yet to be discussed with FDA.
Yeah, excellent. Well, James, I just want to thank you again for coming on board with Sharewise and just doing a little bit of a presentation for your shareholders and anyone else new out there that is thinking about getting an investment in Nyrada. The key topics he's discussed. There's a lot of detail there. There's a lot of plans. There's obviously a lot of time and effort put into it. So it is exciting. He looks like he knows the ins and the outs enough to hopefully execute the plan. But it is a waiting game at the end of the day with some of these things. Is there anything you just want to note just quickly before we wrap things up, James?
So the aim is to provide some hope to these patients and their families, and the hope requires a plan. We believe that we've got a plan and we've got the right people in place to execute on the plan. So we'll leave it there. And thanks, Harry, and thanks everyone for
thank you.
joining.
And if anyone has any questions or queries, feel free to reach out to James or us here at Sharewise. I'm happy to have a conversation with anyone. Just jump on our website. You can fill out a speak to an advisor form on there. This is recorded. James will have a copy. We have a copy on our YouTube channel. So everyone, thanks again for coming and watching the webinar, and stay tuned for more to come.
Yep. Thanks, Harry. Thanks, everyone.
Thanks, James. Bye.